Good afternoon. This is Eugene Krenzel, Chief Scientific Officer of Accelerate Health, is welcoming you to this very exciting webinar on navigating FDA. Thank you so much for joining us today. We have a fantastic crowd. We have more than 400 registrants, and we have terrific presenters today that we'll introduce in just about a second. First, a couple of words about Accelerate Health and Accelerator Network. Accelerate Health is a healthcare-focused early-stage SBA award-winning accelerator for early-stage startups in biomedical and healthcare space, headquartered in Louisville, Kentucky, with a national footprint, having an operation in Michigan, and also having a lead in their accelerator network, along with University of Kentucky as our academic lead, and a great constellation of 24 universities across 6 states and Puerto Rico, working on NIGMS-funded technology transfer accelerator hub. Our guests today, we have 2. The first one is Chris Asiela from National Institute of Health, their Office of the Director. She is Innovator Support Team Lead in the Small Business Education and Entrepreneurial Development Office, called SEED at NIH, where she coordinates activities of a team of a great group of professionals with tremendous experience in product and business strategy, business development, fundraising, partnerships, reimbursement, and regulatory affairs. She's been involved for more than a decade, providing various regulatory support and consultation to academic innovators and small businesses engaged in therapeutic, device, and diagnostic development programs. She has a bachelor's degree in chemistry and PhD in pharmaceutical sciences from University of Maryland. And she is certified through regulatory affairs certification since 2011. Chris, that is now time for you to
start screen sharing.
Start your screen and go with your presentation, please.
Absolutely, happy to do so. Making sure that I am doing this right. Hopefully you are seeing my screen at the moment. All is good?
All is good.
Wonderful. Okay, well, thank you, Eugene, for the invitation to present today to the NIGMS Hubs. I really enjoy speaking with first-time entrepreneurs, academic innovators, and small businesses. That's where I've been sort of my lifeblood for the past 10 years. And so I'm very excited to be participating in today's webinar. And let's see, yeah, there we go. So as Eugene mentioned, I have been in the regulatory affairs world for a little bit of time now. I started out with a fellowship at FDA in the Center for Drug Evaluation and Research, and at the end of my fellowship, I joined a contract research organization, and as a regulatory specialist, grew to be the director of their regulatory affairs and services team. Then I joined the National Heart, Lung, and Blood Institute as the regulatory specialist in a brand new office that was created there to focus on a community much like the one that's in the room today. That experience prepared me, hopefully well, we'll see, to take this on at a much higher level role across all of NIH in the Office of Extramural Research in the new SEED office. And because the office is new, I just want to take a few moments to sort of introduce you to it. I'm actually going to turn off my video while I do this because I'm distracting myself by seeing the video. So we are, as I mentioned, a new office in OER, and we coordinate the activities of the small business program. We provide a space for discussion and dissemination of best practices among academic innovation programs, much like the Accelerate Health Hub that's sponsoring today's event. And we provide support to the NIH innovator community in multiple ways, including through that team of experts that Eugene just characterized. I encourage you to go ahead and take a look at our website, seed.nih.gov, to learn a little bit more about us. but I also encourage you not to judge us by it. We are only 1 year old, and we inherited a bunch of stuff that's been cobbled together. So if you check it out this week and you check it out in a few months, hopefully in a few months' time, you'll be much more impressed. But moving on to the topic of today, which is navigating FDA, I like to have learning objectives in my talks. And so my objective for all of you today is to help you get a high-level understanding of how to figure out how to get your amazing idea through and to FDA and into humans. So we're going to do that by covering 3 primary topics. First, what information can you find on your own? Second, how do you use that information to guide your research? And then 3, understanding when and how to meet with FDA. So FDA has as their primary consideration risk-benefit ratio. This is the driving force behind every decision they make. The benefit doesn't need to be quite as great and exaggerated as is expressed on this slide, but it does need to be greater than the risk in order for you to be able to do what you're asking to do. The degree of benefit is always relative to the severity of the indication or the type of illness, its prevalence, and what other treatment or care options exist. Risk benefit is assessed at every single stage of the product development lifecycle, from first in human to post-market, okay? We talk a lot about the FDA, which does stand for the Food and Drug Administration, which I hope everyone knew, but I have seen people get that wrong. And so FDA is actually a much larger organization than is represented on this slide. However, it's useful to understand where biomedical review happens. And so there are 3 primary centers where that happens: the Center for Biologics Evaluation and Research, the Center for Drug Evaluation and Research, and the Center for Devices and Radiologic Health. There's, as I said, a lot more to this. There's food regulation, cosmetics regulation, veterinary drugs, etc. But these are where we're going to focus today. And I will be focusing my presentation primarily on the Center for Biologics and the Center for Drugs. Ben's presentation will focus on the Center for Devices and Radiologic Health. The Center for Biologics has 3 primary review divisions. Again, the entire organization is larger than this. The URLs at the bottom of my slides will take you to the full org charts, but it's helpful to understand what sort of products does FDA consider to be biologics. So looking at just the overall titles of the offices can be helpful, but sometimes you want to dig a little deeper, and these are the divisions within those offices. One thing that you will notice here is that CBER actually also regulates a whole lot of medical devices, because the medical devices and tests used to safeguard blood components and cellular products takes up a fair amount of space in their portfolio. So you might want to consider, depending what type of technology you are developing, even if you are developing a device, it may indeed be used and regulated by the Center for Biologics. The Center for Drug Evaluation and Research has, in their Office of New Drugs, they recently reorganized. So if you used to see the Office of Drug Evaluation 1, 2, 3, 4, 5, and that was very opaque, no one knew what that meant, they've made it a lot more transparent. It's a lot easier to find where your technology might be regulated. And I want to point that the Office of Therapeutic Biologics and Biosimilars, in 2002, almost 20 years ago at this point, monoclonal antibodies and recombinant therapeutic proteins were classified by law to be regulated in the Center for Drugs. So even though they are technically biologics, they are not regulated by the Center for, by CBER. And again, the URL here allows you to dig deeper into these office structures, understand which division or branch might be appropriate for you to reach out to as you are developing your technology. So that was sort of the super high level tour of FDA. One thing that's interesting about FDA is that it's the only drug regulatory agency in the world that is required to publish its rationale for approving a new product. This is because the US has the Freedom of Information Act, which was passed in 1967. And so the FDA, both proactively and in response to FOIA requests, as we call them, Freedom of Information Act FOIA requests, posts information online to inform the public about the products they regulate and allow into commerce. The importance of FOIA for you as a drug or device developer is that there is a lot of information in the public domain that comes directly from the FDA, right? You get to know what they're thinking about the absolutely newest technologies and drugs that are coming through to the market. And you get this information by looking at approval documentation, advisory committee transcripts. They publish guidance documents, they deliver webinars and seminars. And what we're going to do in the next couple of slides is take a look at some of that information so that you can become familiar with how to find it all on your own. So when you want to learn about regulation, I always tell people if you're looking at FDA, you definitely want to go and look at the FDA.gov website. It effectively offers you about 85%, 90% of what you're going to need to know to understand your market area. You are also, however, going to need to supplement that information with other online resources. You're going to want to look at manufacturer websites. You're going to want to look at contract manufacturing or research organization websites. There are a lot of how-to guides and templates, webinars, blogs, video casts, podcasts, things of that nature that you can find. You take all of that information and you sort of need to think broadly in a Venn diagram, or what is it, the flower of life, all those intersecting circles, right? About how all of this contributes to your personal technology development. You may be developing something in an area that has never had a therapy before, so you're going to have to look at a sort of a sistered up area, a border area. Are you developing a injectable versus a patch versus an inhaled therapy versus a tablet formulation type of thing? So you can pull information, think really broadly as you're doing your research, and then narrow that funnel down to get you to your specific plan for your specific drug, biologic, technology. So we're going to go through 4 different FDA.gov resources today, and the first is looking for information about biologics. So if you're developing a biologic, you have to have a little bit of information already about what you're trying to find before you go looking. You need to know either the year of approval or the name of the drug that you're looking to copy information from, or something similar. Once you have that, you can search the Biologics database and find all sorts of useful information. So I try and keep URLs on the page when I take screenshots if I can, to allow ease in you guys following along once you get the deck. And so today I'm going to take albumin and follow this through. Okay, so I clicked on that albumin link on the prior page and I got to this page. And as you can see, it gives some information about the license number, who makes it, things like that. You can look at the package insert. And then you can take a look at the supporting documents older than 3 years, because this product has been on the market for more than 3 years. This information is going to help you understand what sort of information was used to originally get this product to market, and any innovations or label claim expansions that have come since it was originally on market. Let's go ahead and click through this link, and what we get to is this page. Now on this page, I want to draw your attention down here to the bottom. You might have thought I was going to go to that approval letter, but indeed that's not necessarily the best place for you to look. That's just a letter. It's basically a form letter with plug and play information. What you want to look at is the summary basis for regulatory action. And that will give you category by category the reviewer's comments and analysis of the test results and the data that Octapharma, in this case, submitted to FDA to demonstrate safety and efficacy of their albumin product. So if you click through that, This is the first two of, I think it was maybe 8 pages of information. Sometimes, and this is old, this was back in 2006, right? So some of the newer documentation is much more extensive, is broken out by specific review discipline, but this is a general format that you would see, right? So you'll see information and it will be redacted. You'll see places where things are blacked out or otherwise removed from public disclosure, because even though there is the Freedom of Information Act, that doesn't give anyone the right to know trade secret information, right? So as you're developing your drug, the things that are important, imperative, and very specific to your drug will still be protected. Nonetheless, you can take a look at this information and begin to understand the types of testing that you're going to need to do, start developing your plan, and understanding how to guide your own research, right? Let's go back to that same front page for albumin, right? And take a look over here on the side. We have this pre-market approvals and substantially equivalent 510K device information. Now that has nothing at all to do with the fact that this is albumin, right? This is not related to the drug product. This is just the left-hand navigation bar. But what is this? What are these things? This is where, if you remember, I mentioned the device information comes in, right? So CBER regulates a lot more medical devices than you might think. And if you're developing a medical device that you can't find any information about in the CDRH, or the Center for Devices and Radiologic Health databases, chances are you should come take a look at the Center for Biologics website before assuming that it's unregulated, right? But again, I'm not the one who's an expert on medical device regulation, so this is as much as I'm going to do is raise a flag of awareness, and I'll let Ben talk about how to do that research later on during his talk. Another really great resource to learn about FDA regulation is the Learn website for each FDA center. These websites are where they post presentations, webinars, information for the industry or public, things that might be useful to better understand what FDA does, why they do it, the way they do it, and how they think about things. These pages are really, really useful. I always encourage early stage innovators to go take a look and learn about what it takes to file an IND, or early phase trials for cell and gene therapies, right? Whatever it is that you're looking for, chances are greater than 50% you will find something on a Learn page that is useful for you. Let's move on to the Center for Drugs. Just like I started with the biologic licensed and approved products for small molecule drugs, there is this page, which is drugs at FDA, right? And here I would like to identify some information about a fairly common pain relief medicine, but I can't remember exactly how to spell the name of the generic ingredient. So I've only put in a few letters. And that's okay, because look, it found it. It found ibuprofen based on ibuprofen, right? It gave me exactly what I wanted, got me to the right place, and this is page 1 of some number of pages. And doing this, now I'm able to click through, and I'm going to choose Advil. And this Advil page, the link brings me to a page that has all kinds of useful information. Again, I can see how the drug was originally approved, has labels changed over time. And if I click on the approval dates and history, letters, labels, reviews, etc. Page, it will bring me to documentation very similar to what I just showed you through the Center for Biologics approval site. It will include information about chemistry, manufacturing, controls testing, preclinical and non-clinical testing, the number, size of clinical trials, the types of animal models that were used, the types of release testing that's expected, those sorts of things. For a deeper dive into this sort of research, I encourage you to watch another webinar that NHLBI developed several years ago called Developing a Target Product Profile. That particular webinar goes in depth on how to research this type of information for drugs and walks you through the process sort of step by step. And just like CBER, The Center for Drugs also has a Learn page. That page has tons of useful information. The Center for Drugs actually has a whole lot more information than the Center for Biologics. It's a larger center. There are more people there. There's more people in this office. There are also a lot more products regulated. So they really want to push the information out into the public domain because that helps you gain access to their insight faster because you don't have to actually wait to talk to one of them. But you still, of course, can. This is not a problem. You can always reach out to FDA if needed. If you take a look through some of these things, you'll see there are podcasts you can listen to while you're going for a walk in the afternoon, or you can watch a webinar while you're at lunch, read through a slide deck, lots and lots of topics, tons of great info. I encourage you to check it out. And then, of course, as I mentioned, internet searches are great to supplement all of that. And as you take a look at some of the examples on here, you'll see webinars, you'll see access to mentors, you'll see guidance, assay development guidance manual, clinical trial templates, and IND templates, and all sorts of other great stuff. It's out there on the web. Be creative in the way that you search. The possibilities of what you'll find are limited only by your imagination as you create search terms. So hopefully that was a pretty good summary of how you can extract some really useful, pertinent information from the public domain to help you understand what the regulations are. But now you need to take that knowledge and use it to guide your research, right? So how do you do that? I'm going to, in this case, pull up what is called a guidance document, an FDA guidance document. And this is the guidance for current good manufacturing processes, in case you don't speak acronyms, for phase 1 investigational drugs. And this is published by the Center for Drug Evaluation and Research. And as you can see from the table of contents, it goes pretty well through all of the things that they would expect you to provide them, all the types of data they would expect you to provide when you want to put a new molecular entity into a human being for the very first time. It describes their expectations in multiple disciplines, and that information can be used by you to develop your research plan. So let's start with chemistry, manufacturing, and controls. Based on that guidance document, you would read through it and you would understand that you need to have a plan to develop specific, sensitive, and accurate assays that are going to assess the purity, potency, identity, and strength of both the interim and final materials that you want to put in humans. So the manufacturing process interims, as well as the final finished product, right? Prior to this, you may have thought you just needed purity and potency, or purity, potency, and identity. And now you know you also need strength, right? So reading through these documents helps you understand what you need to include in your certificates of analysis. And the research plan would be how you're developing the work to be able to build that final document, right? And I hate to read my slides, so I'm not going to read through all of this. Hopefully you've been able to read it while I'm chatting. Okay, the same thing would go through with preclinical research to inform a regulatory plan. However, the difference here is that much of the preclinical development of new drugs has been harmonized through the International Council for Harmonization, okay? And this is a truly international body that is global in scope, and these guidelines are based upon best practices. They have been agreed to by industry, regulators, and patients in terms of developing the best possible outcomes for human trials and developments, right? And these guidelines fall into 4 categories: quality, safety, efficacy, and what is called multidisciplinary, sort of like the other tab that you would see. And if you come to the safety, that's where you would find out information about preclinical expectations, okay? So you would go through here and you would take a look at these different types of guidelines that are in here, and you might, from that information, understand how to build a research plan that will validate the development or validation of your animal models, making sure they're appropriate for your therapy, understand how to truly build an absorption, distribution, metabolism, elimination, and toxicity profile, or ADMET studies, right? Understand how to initially evaluate and propose dose ranging studies, and understand for your specific drug, are you going to be giving just a single dose? Is it going to be a repeat dose? Is it going to be a chronic dose? How much information do you need before you go to a regulatory body and propose to do some first-in-human studies? Okay, so again, all of this information is available online. It's great for you to be able to go in, take a look, learn from it, and use that to help guide your research. But at some point, you've got to meet with FDA if you're developing a drug within the US. All right, you've done your homework, you've made your plan, and you think you're ready to move forward, but really what's happening is you need to validate your plan before you go ahead and you spend $250,000 on a 28-day rat tox study, or $500,000 on your large animal IND-enabling studies, right? So as a best practice, and particularly for first-time entrepreneurs, first-time drug developers, it really is best to validate your plan with FDA. So, FDA can be a scary place. You may not know how to do this, and you may just have some really basic questions, think you're not quite ready. If you're at that point, if you don't think you're quite ready for a meeting, what you can do is you can reach out to the offices of communication within each of the centers. And I've listed them all here on this slide, but they're also just very easy to find online on the FDA website, contact us sort of things. You can send them an email, ask for basic information. If you're asking something too specific, they'll hook you up with a review group. They won't answer it. But if it's general information, you will get a response from these offices. Generally within 72 hours, you get an initial response, and within 14 days, you get a full response. That full response may come at 72 hours, but if it's a complex question, they may take some more time. Before you get to the human study aspect of your product development work, there is the possibility for earlier stage meetings. With the Center for Biologics, they really do like to encourage people to come in for what they call an interact meeting. This is informal, it's a non-binding regulatory consultation, specifically initiated in the Office of Tissues and Advanced Therapies back when they had a different name. It's meant to be an early development meeting to help guide your development of both the manufacturing processes as well as your animal studies. And this is highly encouraged for people working in the cell and gene therapy space. For the Center for Drugs, you can request a consultation meeting. It's similar in nature to an interact meeting. It's still that informal early meeting space, non-binding. It's not available through all of the offices or divisions, and it's typically only granted for super novel products where there isn't really a history of a lot of experience in that regulatory space with either that molecular class, that metabolic profile, or that disease state, okay? But remember, at some point you are going to want to meet with them. So how do you know when you're ready to meet with the FDA? You have some minimum number. I usually go with 3 as a minimum number and about 6 as a maximum number of critical questions. Those questions should be ones that can only be answered by FDA, right? You or a knowledgeable regulatory professional cannot answer them. It's new space. And if you can't get those answers, your work is going to stop. So again, with this concept that FDA does everything as a risk-benefit ratio, there are 3 types of meetings. There are urgent meetings, which are type A, important meetings, which are type B, and meetings that still need to happen, but are neither urgent nor super important. So those are the type C meetings. They're anything that's not A or B, right? And so typically, most innovators will start by trying to plan for a pre-IND meeting, and these are type B meetings. These are requested to discuss some sort of planned developmental milestone. There are typically 3 formats for the meetings: face-to-face, virtual, and written response only. In light of the current global health crisis, there are no face-to-face meetings with FDA. Everything is either virtual or written response only. There are timelines for the FDA to respond to you, and the parentheses around the 50 days before the scheduled meeting date for submitting a briefing packet is specifically for end of phase meetings. Lisa, I believe you may be unmuted.
Sorry.
And then the type A meetings are generally, they're not always, but they're generally clinical plan is on hold. Patients have been exposed or patients would be exposed to the therapy and there's a problem. FDA has a problem or a concern. So they put you on clinical hold, and then you need to put together your response and request a meeting to discuss your response with them. Again, there are timelines associated with this. So what goes into that briefing packet? You need to submit the information to FDA for them to understand your questions, understand your response to their questions, and for them to have internal meetings before you meet with them, right? So you provide a briefing packet. It includes all of that background information, including any questions, whatever. FDA internally meets to talk about it. They agree upon how they're going to respond to you. They provide preliminary written responses about 2 days prior to the meeting. Sometimes rarely it's earlier, sometimes it's a little later. You then take those responses and you decide you still want to have the meeting. If they said, yes, we agree with everything you're proposing, then you may not need to have the meeting, right? So you have that option at that point. Meetings are only granted for 60 minutes. They're chaired by FDA. The conduct of the meeting should follow brief introductions, focused conversation around those areas where FDA's response was not in agreement with what you proposed. And then at the end, a brief summary of the discussion and agreements that came out of the conversation. The sponsor or you provide draft minutes to FDA. FDA then finalizes the minutes, adds them to the official file within 30 days of the meeting, and sends them back to you. So as I mentioned at the beginning of my talk, I had 3 objectives, and I hope I met my goal. We're going to find out during the Q&A session momentarily, and if you have any questions after this talk is over, please feel free to reach out through any of these mechanisms. Thank you very much.
Thank you so much. Chris, could you unshare your screen, please? Wonderful. Okay, so if you guys have any questions, please either raise your hand or type your question in the chat, and we'll call on you to voice that question. So far, we had questions about the recording of that webinar. Yes, it is being recorded and will be placed on Accelerator Network's website. I actually have a question while we're waiting for others. Chris, thank you so much for a fantastic presentation. What would be your recommendation to researchers, not necessarily to people who are already in the commercialization stage, but who are still doing their still basic research, but what do they need to think about as they're doing that basic research on their future FDA strategy, and what would be a good source for information for them to learn about what they will need to take into consideration down the road?
Right, so that's the most common conversation I have with people, and the resources I point them to are exactly the ones that I included here. So depending where a team is in understanding their drug development effort, if you are still at the point where you are identifying a single lead, then your most important conversation is going to be with your medicinal chemist, for instance, right? Understanding the physiologic properties of the molecule, what's going to help you get the, the best formulation of product so that you can get greatest biodistribution or most targeted distribution throughout the body, those sorts of things. And as you're doing those things, if you read through the types of guidance documents and documentation that go to that first in human exposure, so that current GMP for phase 1 investigational new drug guidance document that I put up there, I chose that very specifically because there's a lot of really critical information in there that talks about the quality attributes. And many times the scientists that NIH funds are very interested in the biological impact of the molecule that they're studying. And they kind of forget that if you do certain things in the chemistry and manufacturing processes of that, that is, for many NIH innovators, a primary reason for being put on hold when they first submit their IND. So don't neglect all three parts of the stool. You need a manufacturing process, you need the preclinical safety data, and you need a strong and appropriate clinical plan.
Thank you. Question from Linda Dwoskin from University of Kentucky. Linda, you can unmute yourself and ask the question if you would like to do that in your own voice.
Sure. I was wondering if you could explain a little more about what you mean about binding versus non-binding from the FDA response.
Sure. So FDA, a non-binding FDA response or meeting is one where there is a discussion and FDA is saying, we think this is the best way for you to proceed, but you can choose to say no, right? If FDA says you should demonstrate in this, in this animal model, or you should perform these types of analytical studies in order to show something to us, we think you should do that. We think that would be smart of you to do that because we have that question. You can still say no, it's not binding, right? And if you come back with some other method or some other model, they will evaluate what you give to them. A binding agreement with FDA says that if you do exactly A, B, C, D, and present them the data, that as long as you meet these pre-specified targets, let's say you are going to, I'll bring it to a very clear example. They propose that you are going to do a pediatric study in children from 2 to 10 years old with your drug, that you will enroll 3 cohorts. Your first cohort will be 50 children, your second cohort will be 30 children, and your third cohort will also be 30 children. So you have 1 active arm, 1 comparator arm, and 1 placebo arm, right? And if you do that study and you analyze the data and you come up with a p-value of 0.05 in your result based upon your proposed endpoints, right? If all of those agreements are met, then FDA will agree that that is sufficient and they will be bound to that decision.
Okay.
But that's not normally how it works. Usually it's best understanding of the science at the time with good faith guidance of what they would like to see. And you are a scientist, you are a brilliant person in your own right. You are allowed to come back to them with something different.
Okay.
Thank you.
Thank you.
Yep. Tamer Mohammed, you can unmute yourself and ask your question. Tamer Mohammed.
Yes, sorry. So I just, if you can comment more on how much evidence do you need to have in terms of efficacy and safety before, to show the FDA before you schedule the meeting on? Do you have to have at least animal studies done? Do you have to have, what kind of evidence that you need, at least just giving examples?
Okay, so that's going to depend on a number of different factors. So if you're developing a cellular gene therapy, that amount of data is somewhat less than it would be if you're developing a small molecule, right? If you're developing something for a very common disease or disorder, you're going to need to provide them with some more data than if it is for a rare disease where there is no current therapy right? Where the risk is that there is nothing. And so the risk benefit ratio then goes to, well, there may still be high risk in trying this, but there might be benefit. And so we'll go ahead and, you know, have that conversation. It's really extremely project specific. I tend to encourage people to go not so much by how much data do I need, as I mentioned in my talk, it's more about what questions do I have. So the questions can be, is this sufficient data for me to move forward, right? You don't have to go in and say, I have all of this data and now I'm going to do, I mean, you do go in saying that, but you can come in and you can ask the question, is this data sufficient for me to go from small molecule to, or, you know, from a small animal model into my pre-IND or my IND enabling studies.
Thank you. We have time for one more question before we go to the next presenter. Parajad Bhatnagar, would you please unmute yourself and ask your question?
Hi, Chris. Thanks for the presentation. My question was in context of cell and gene therapy. Like we have been able to generate some efficacy in some time and points left on our NIH grant. I want to maximize the productivity from this grant. What data do you think we would suggest we can collect beyond efficacy that will make our approach an attractive approach for FDA to consider? What kind of data would FDA ask for?
Okay, so your audio wasn't 100% perfect, but I think I understand the question both from what you wrote and from what I was able to hear. So if you are developing a cell and gene therapy, there are a number of guidance documents that you should really take a look at. There are unique aspects to many times the initial therapy is developed in, for instance, a murine model and then moved into using murine genes and then moved into a humanized system using humanized genes and then creating the animal models that will be able to demonstrate efficacy and the development of autoimmunity or responsive immunity to the agents themselves, things of that nature. There are some really, really excellent guidance documents on that, and the development of cell and gene therapies is actually something where you can request what's called a specific designation, RMAT designation. Forget exactly what that stands for. But one of the things I think that's really important in terms of basic awareness for cell and gene therapies is to know your genetic sequence. So as you're developing your plasmids, knowing that they're clean, knowing that you really do have a single clone that you're populating, developing, you're working in your master cell banks. These are the types of things where if you're focused on just the biological effects and you forget the chemistry, manufacturing, control aspects for cell and gene therapies, that is the product. It really is all about the manufacturing, ensuring that's clean, you're mycoplasma free, et cetera, et cetera. So that sort of thing becomes very, very important. It's also important to recognize that most cell and gene therapies are done in essentially a protein-free environment, certainly animal serum-free environments. So you want to identify how you're going to transition your target therapy into an actual therapy and start thinking about the complexities of that, that technological transfer from the research grade material to potentially human grade material.
Yeah, we have one quick question from Dr. Nancy Gua from WVU.
Nancy's question is not quick.
It's not. Okay.
That is a whole nother webinar in and of itself.
Okay. Would you like to answer somewhat quick now, or should we see if we have time at the end of both sessions before we go?
So I think let me try and do this quickly, and then if Nancy wants to follow up with me specifically and uniquely afterwards, we can dive into that. So CLIA versus FDA approval. CLIA is the Clinical Laboratory Improvement Act. It is not regulated through FDA. It is regulated through the Centers for Medicare and Medicaid Services, or CMS. And it is oversight of clinical laboratories, right? So clinical laboratories can develop their own sets of tests, and those tests do not need to be, although they frequently are, regulated and cleared through FDA. So they're depending on the particular type of diagnostic assay you're looking to develop and where you're looking for it to be tested. If you want it to be tested just in one lab, where everyone sends their samples to a central lab, versus sending kits out to many labs and allowing it to be tested in multiple locations, there are variations on how you would develop your assay and validate it. But that, like I said, that's a whole nother webinar. And so, Nancy, feel free to follow up with me. The secret sauce to contacting people at NIH is first name dot last name at nih.gov. So anytime you see a NIH speaker, know that that's how you can get a hold of them.